Good afternoon, ladies and gentlemen. My name is Niamh Bambrick and I'm the president of the Agricultural Science Association. I'm delighted to welcome you all to the first part of our webinar series today. So today we have Dr. Siobhan Walsh, who's going to chair the webinar for us. And then speaking, we also have Dr. Ewen Mullins and Dr. Helen Sheridan. So just a bit of housekeeping. Everybody who I suppose is registered for today's webinar, you'll be automatically registered for Thursday's webinar, and you will get an email about that later on today. And then we also have the Q&A function in the Zoom. So if you just click on the Q&A function, you can type in a question to any of our panelists and they can see it as the webinar progresses. So without further ado, I'll hand over to Siobhan.
Thanks very much, Niamh. Okay, so today's topic is adapting tillage and forage production systems to a changing climate. And we have two top-class speakers, Dr. Helen Sheridan and Dr. Ewen Mullins. So Helen is going to start us off. Helen is a lecturer in UCD in farm landscape ecology and agri-environmental policy. And Helen, will let you start to share your screen there. As Niamh said, please remember to put the questions into the question and answer box below.
Great, thanks for that, Siobhan. Can you see that okay?
Yeah, I can see it, Helen.
Great, thanks very much. Okay, well, I'll get going with it. So today I'm talking about work that has been completed by ourselves at UCD and also our collaborators in Teagasc and elsewhere. I suppose if we think about our agricultural systems, food production is obviously key to farming, and we all need to eat at least a couple of times a day, so that's not going to change in the future. However, I suppose things that have changed quite significantly over recent decades is this idea that we have a greater appreciation and understanding that our farmed landscapes and therefore our food production systems are not just about food production. We also need the delivery of a whole range of other services and functions from that landscape. And how we manage our landscape is going to impact on how well it can deliver those services and functions. And how well those services and functions are delivered will inevitably impact on our levels of food production. So I guess, in terms of this, food security is fundamentally underpinned by our environmental sustainability. I suppose that increasing understanding of those relationships, although there's many we still don't know that much about, but we've seen that there's been an increase in the focus in terms of policy and legislation in terms of our environment over recent years. Now, we know that farmers have to deal with already a range of directives, legally binding directives, in terms of their commitment to the agri-environment, so the nitrates, the habitats, the birds directives, etc. But in more recent years, we have seen, the development of documents, strategy documents and agreements like the European Green Deal and the Farm to Fork strategy, which within those, we now have targets of reducing our fertilizer inputs by 20% by 2030, for example, while at a national level, of course, from an agricultural perspective, there's a target there to reduce our greenhouse gas emissions by 25% by 2030. So I guess in terms of achieving this food production into the future, we're going to have to do that against a backdrop of having reduced inputs, increasing our efficiencies and also reducing our impacts on the overall environment. And I guess given that we know that we are seeing a degree of climate change and will continue to see a degree of climate change, we're going to have to show enhanced adaptation and resilience within our systems in order to be able to sustain our food production. So I suppose the piece of work that I'm going to focus on is the research that we've been doing around our grassland swards and what we've been finding out there in terms of when we add a little more diversity into those systems. And I have to go back, it's about a decade now since this work was done, undertaken at UCD Lyons Farm, where we looked at a whole range of different mixes. So when I talk about mixtures here, I'm talking about mixtures of grasses, legumes and forage herbs. And we can see along the right-hand side of the graph there a legend that gives us the breakdown and the seeding proportions of those. So, for example, the yellow line represents a mixture that contained 40% grass, 30% legume and 30% herb when it was sown. On the x-axis, we're looking at increasing nitrogen fertilizer application rates from 0 to 135 kgN per hectare per year. And on the y-axis then, we're talking about kilograms of dry matter yield per hectare. And I suppose this was one of the first pieces of work that we did in this space. And what was really interesting and exciting about it was, when you look at the dotted black line there, that's our yield in terms of dry matter production from our perennial ryegrass monocultures that are receiving 250 kilograms of nitrogen per hectare per year. So when we look at this graph, we can see that a range of the different mixtures actually perform really well relative to the ryegrass monocultures, even when they're receiving 0N. And when they actually receive about 90 kilograms, they're overproducing, or they're producing more than the ryegrass monocultures were. So this was a really exciting find, I guess, in terms of the research that we were undertaking at Lyons. But of course, that was undertaken at plot scale, and we needed to see how swards would perform under grazing. And this then became part of Connie Grace's thesis, where she looked at 4 of our sward types. So a high N, or a relatively high N input perennial ryegrass monoculture, ryegrass and white clover, a 6-species mix and a 9-species mix. And she looked at those under grazing of ewes and their lambs. And what was the finding of that was that when we compared the lambs fed on perennial ryegrass to lambs fed on any of the other mixed sward types, so ryegrass and white clover, the 6-species mix or the 9-species mix, we found that the lambs finished about 2 weeks earlier. They reached their slaughter weight about 2 weeks earlier when fed on any of those more complex diets than ryegrass. And that has very significant implications when you think about the necessity to reduce the lifespan of our livestock, or reduce the time to slaughter. Maybe more excitingly even was this work again by Connie Grace and Tommy Boland, where we looked at the effects, or they looked at the effects of the sward types on animal health. And in this case, in the worm burden within lambs. And here we're showing the number of anthelmintic treatments that were required by those lambs. And this is where you see the multi-species sward, so a 6-species mix and a 9-species mix here, made up of grasses, legumes and forage herbs. We see those requiring about 50% fewer drench administrations in terms of the lambs that were grazed on those relative to the ones that were grazed on the perennial ryegrass monocultures. And again, that becomes hugely important when you think about the extent of resistance that's building to the existing active ingredients in terms of treating worms within livestock. So I've been talking about mixes for a few moments now, and I guess when I'm talking about that, I've talked about grasses, legumes and forage herbs, and we had various different types of mixes during that work at Lyons. But the one that we found to be our best performing, and I'm not saying it's the best performing everywhere, but certainly under Lyons conditions, was this 6-species mix of perennial ryegrass and timothy as grass species, white and red clover as legume species, and chicory and plantain as forage herb species. And what you'll notice as you look at those really is what we're trying to do when we select those different species from the different functional groups is to maximize the kind of differences between them, so that we have shallow rooting species and deep rooting species, for example, but also species that grow to different heights and what have you in the sward. And the idea is that, you know, when you grow species in a mix like that, that they will be better able to use available resources than one species grown within a monoculture. In addition, that we get synergies between species. And when it comes to kind of reducing inputs and increasing efficiencies, understanding these kind of relationships between the plants that we put into our swords becomes increasingly important. And it's apparent that we need to develop that understanding. So that led us then to the establishment of what's become known as UCD long-term grazing platform, and that's based at Lyons Farm. So we took that best performing 6 species mix that we had identified in earlier work and established it in a farmlet at Lyons, together with 2 other farmlet types, a high-end input ryegrass monoculture and then a ryegrass and white clover farmlet also. So we had the third one was our multi-species mix. And we had 8 hectares under each of those farmlets, and they were stocked at 2.5 livestock units per hectare in a dairy calf to beef system. And in terms of sward performance, when we look at that, we can see, and we've found consistently over the years since this was established in 2019, that our multi-species swards, when compared to our perennial ryegrass ones, will have produced 2.5 tons of herbage dry matter additional to what's been produced on the ryegrass monocultures. And the really exciting part of this is that they're only receiving 44% of the fertilizer nitrogen that the ryegrass swards are. And of course, you can see that the ryegrass and white clover are sitting in the middle in terms of dry matter production. So, of course, producing the herbage isn't enough. We've got to see how animals perform on it. And certainly the animals to date, they have a target slaughter weight of 620 kilos. And we've seen that when we move from the ryegrass into the other sward types, towards the multi-species, we're losing 35 days on average of their lifespan. So there's generally a week or 2 between the ryegrass white clover livestock and the multi-species livestock, but between the ryegrass monocultures and the multi-species, we see a 5-week gap between those animals reaching their target slaughter weights. Again, hugely important when you think about, the MAC curve and what's required to achieve our emissions reductions. So taking that into account, coupled with the reduction in nitrogen use on these swards, we can see that as we move from the ryegrass again over the different sward types, we're seeing about a 14% decrease in the CO2 equivalents per kilo of beef carcass produced. So a really significant reduction in the carbon footprint of those systems relative to the ryegrass monocultures. It's not alone at Lyons that we're seeing this. Certainly we've been involved in another project at the Devonish lands at Drought, where we had a similar but different farmlet experiment up there, where a high-end input perennial ryegrass monoculture. In this case, we had a permanent pasture, a 6-species mix, which was similar to the one in Lyons, but then a 12-species mix as well. And in that case, the swards were grazed by cattle and sheep, so they were co-grazed. And again, in terms of herbage production, we're seeing that 2.5 tons extra herbage dry matter production from the 6-species mix relative to the perennial ryegrass monocultures. And again, only receiving 41% of the fertilizer nitrogen in this instance. So certainly it's happening across different sites, and it's well established at this stage that we can produce more using less in terms of herbage dry matter production when using these more diverse swards. In terms of the animal performance at Drought, this is the lamb findings here, and the lambs grazing the 6 species and the 12 species mixes had 30% and 21% increased average daily gains compared to the ryegrass and the permanent pasture swards. And again, similarly with the heifers that were on those different sward types, we see significant benefits of moving towards the 6 species mix in terms of average daily gains there. So we know that the swards mixtures have the potential to grow more. We're using less N when you're producing that, but then we have to, we've talked about the methane and the carbon dioxide equivalents associated with the system in lions. But this is some work undertaken by Saoirse Cummins with my colleagues in Teagasc, Johnstown, and with John Finn down there. And you can see on the left-hand side of your screen there, different swards that were being treated with different levels of nitrogen. So perennial ryegrass receiving 300 kgN, perennial ryegrass at 150, and then a 6 species mix also receiving 150 kgN. And on the y-axis here, we're looking at nitrous oxide emissions per tonne of dry matter yield produced. So Saoirse took the yields from these swards, but also looked at the nitrous oxide emissions. And you're seeing a very significant reduction in terms of the N2O emissions associated with each tonne of herbage produced from her research. On the right-hand side, again, just follow up on it, this is a heat map which shows on the different vertices of the triangle here, a sward type we'd say at the very top would be 100% grass monoculture. On the left-hand side would be 100% legume, and on the right-hand side 100% herb. And in between we've got various different mixes of those functional groups. And as you can see, as the colour gets darker, the nitrous oxide emissions are increased. So increased emissions associated with that type of system. So we get the highest emissions associated with the legumes and the grass monocultures. But where we have mixtures, we see that we get much reduced emissions, and that's also associated with higher yields. So they have a very important role to play in terms of reducing our emissions of nitrous oxide. Of course, we know that climate change is happening and will continue to happen. It's a case of how well we can adapt to it and how much we can prevent it from accelerating. But, we are going to have to adapt, and we're seeing changes over recent years in terms of drought conditions or also flooding conditions. So it's an important question to ask how these grasslands could potentially react to those kind of conditions. And again, this is work undertaken by my colleague John Finn in Teagasc, Johnstown Castle, where he was working with Switzerland, with Agroscope in Switzerland, and they were investigating, again, 4 different species here, but different functional groups and those different traits associated with the plants. So shallow rooting versus deep rooting species included within their mixes there. And what they're finding was, if you look at the left-hand side, you see of this graph, you see that we have the yields under controlled conditions. So the first line represents from 1 species or a monoculture, then we have from 2 species mix, and then from a 4 species mix. And what we're seeing when we compare that to the right-hand side of the screen there is that the yields from a mixture grown under drought conditions was still higher than the yields from a monoculture grown under control conditions. So it really reiterates this point of the importance of having these different functional traits, which is shown by the plants that we've included in these mixes. And that then confers resilience into the system. It allows the plants to be able to withstand longer periods of drought. They may not be able to get us out of a very prolonged drought, but certainly they don't suffer as much as monocultures appear to. In terms of that work, again, the ability to mitigate against drought conditions, this is Geelong Grange's work, again at Johnstown Castle. And on the left-hand side of your screen there, you see the first heath map. Again, you're looking at monocultures of legume, herb and grass versus mixtures illustrated at the different points within that triangle. And what you can see is under controlled conditions, under rain-fed conditions, we're getting highest levels of dry matter production from the mixes. And that's illustrated through the deeper colouring there, the deeper green. However, when we look at the right-hand side of our screen, this is where there was 9 weeks of drought imposed on these plots. And again, you can see here that it is the monocultures that are suffering greatest when there are drought conditions, while the mixtures are able to withstand. And while yield will decrease, they're able to continue to produce much better than the monocultures can. What does all this then look like when we, see it in operation under a grazing system? And I've taken this from the work at Drought, Jane Shackleton's work at Drought. And this shows growth rates. So it's herbage growth rates per sward type that we had out there. And what's interesting about this growth curve is that you'll see the blue line represents the permanent pasture, the red represents the ryegrass, and then our two mixtures are illustrated by the yellow and the green. And we can see that during the drought period in 2020 that occurred between about March and June, we had quite a prolonged period of drought there. Yes, the yields from our mixtures did drop off, but not nearly as much as the yields from either of the other sward types. And they certainly recovered. They all recovered very quickly once drought ended, but the yields of the mixtures were significantly greater at that time, or the growth rates, I should say, were much higher from the mixtures at that time. So I suppose that's what it might look like on a farm, at farm scale. On the other side of the coin, then, we also know we have significant flooding issues and likely to have more flooding issues associated with climate change. This graph here doesn't necessarily make a direct link with that, but what it does show is that when we put legumes into our mixes, we get greater abundance of earthworms and greater earthworm activity. So if we get greater earthworm activity, we're getting greater levels of pores generated and channels generated in our soils. And what we've found when we look at those different mixes again is that we have higher water infiltration rates associated with those sward types then. And the two are very positively correlated. So that activity between the earthworms producing a lot more channels in the soil and therefore increasing water infiltration. Now, it's not saying that we can alleviate flooding or anything in this way, but what it does indicate is that there's potential there to help water movement within the soils. So it could certainly be beneficial from that perspective. So in terms of the summary of our findings from the work to date, we've seen that we have an increase in herbage dry matter production when we move from monoculture to mixture, and that's been in the order of about 25%, and we're seeing that consistently. Decreased requirement for fertilizer nitrogen, which is in line with what we need to be doing. So a reduction of 60% approximately in order to maintain those yields. We're seeing enhanced animal performance, so reduction in their days to slaughter of 35 days. Improved animal health, so 50% reduction in the use of anthelmintics associated with the lamb production systems. We're also seeing reduced greenhouse gas emissions, the methane emissions because of the shorter lifespan of the animal, and also the nitrous oxide emissions associated with the reduced requirement for nitrogen fertilizers. We've also seen that there's, the quite good adaptation to climate stresses associated with these types of swards. And while I didn't show it here, as you can imagine, there's enhanced economic performance due to the reduced requirement for fertilizer inputs. In terms of some of the current and future work that we're doing in this space, looking at the kind of best composition of mixes to use to adapt them to local soil conditions and local environmental conditions. I've talked here about a 6-species mix, but that mightn't necessarily suit everyone or suit every soil type. The optimum grazing management for them, we know very well how to manage ryegrass monocultures and ryegrass white clover swards, but we're not nearly as sure what the optimum grazing management is for mixtures. We know that we tend to lose the herb component over time out of these swards, so knowing how to rejuvenate the swards with the herb component is going to be a hugely important part of this story. How low can we go with the nitrogen fertilizer is a big question that we need to ask. We've reduced it down by about 60%, as you saw in the work so far, and we're keeping those yields up, but can we go lower? And I suppose that's one of the questions that we're asking with the Department of Agriculture and DERA-funded Multi for More project. We also need to get greater insight into the greenhouse gas emissions and the biodiversity support value associated with these swards, as well as the water quality. And that's all work that is, much of it is ongoing at the moment. So, we'll have a better insight into all of those areas in coming years. So at this stage, while I was the person speaking today, I want to acknowledge all of the people who have worked on this. I hope I haven't left anyone out there, and also to our funders and partners, collaborators on this research to date. Thanks very much.
Thanks very much, Helen. Really, really good presentation. While Ewan is getting ready there, I might just ask you a few questions. So sure. On the 14% reduction on the greenhouse gas emissions, so it was mainly nitrous oxide and then methane from the reduced time to slaughter. Do you know anything about the methane produced while the animals were grazing?
Yeah, so Tommy Boland's been working on that with the green feed units on the animals in the different swards. Okay. So we haven't seen, to the best of my knowledge, we haven't seen any difference between the methane at grass, the methane produced at grass from the livestock. So it doesn't seem to be impacting there. Where it's kicking in is that better performance of the animal and therefore the reduced lifespan of the animal. And of course, the nitrous oxide emissions then as well.
Yeah. And on another one just caught my eye, the 50% reduction in drenches. Like, that's massive. It's so positive from an environmental resistance and an economic point of view as well for the farmer. Those swards were they high in herbage, or how many years had they been sown, and does that matter to the reduction? So if you have a sward that's 3 or 4 years old and some of the herbs have gone out, how does that affect that number?
Yeah, so I suppose that's something we need to see more of. That was on one study undertaken by Connie Grace.
Okay.
So certainly the herbs do decrease over time, and it is the herb component, we believe, that's impacting on that. Well, actually, it's the herbs in the mix together with the other species that's driving that reduction. Now, whether it's secondary compounds in some of those species that's doing it, or maybe it's partly associated with how we graze those swards, but it may be that the animal is just innately more resistant to picking up the worm burden. Still unsure of the mechanisms there, but certainly the herb component is key to, or appears to be key to, having that reduced anthelmintic requirement there. So this is what makes the rejuvenation piece really important, and we have been working on the rejuvenation piece over the last year in Lyons. So trying to get those herbs back in with minimal disturbance to the existing sward, and it seems to be quite possible to do.
Okay, definitely positive there. We look forward to hearing more on that, Helen.
Sure.
So get more questions into the Q&A box for Helen. And you can put some questions in for Ewen as well. So Ewen Mullins joins us now. Ewen is the head of crops research at Oak Park. And Ewen, you have a particular area of research in biotechnology. So we look forward to hearing about that now.
Okay, great. Thanks, Siobhan. Great to be here. So I'll just share the screen. Okay, you should be able to see that now. So, and you can hear me okay, Siobhan?
Yeah, that's working okay, Ewen.
Perfect. So basically, obviously, tillage sector, we're all very familiar with the trouble and the challenges it's had, especially this autumn in terms of putting varieties into the ground. But once those varieties are in the ground, the key thing is to adapt the management and ensure that you can mitigate the risk. And what are the primary risks we have in our tillage sector at the moment are fungal diseases, and indeed other things such as grass weeds. So we have diseases such as septoria of wheat, as you can see here on the leaves. Let me just run the laser pointer, and this effectively can destroy the flag leaf, which is the main engine in a wheat plant, producing those carbohydrates to fill the seed, and will dramatically reduce yield if not controlled. Then we move on to other diseases such as rhynchosporium in barley, ramularia in barley, and of course late blight in potato. All of these diseases will have a dramatic impact, and farmers have to adapt their management systems to ensure that they get whatever plant protection products, fungicides, etc., out onto the crop, so that it's matching the growth stage of the crop that is most vulnerable to the disease itself. When we talk about climate impact, BYDV, or barley yellow dwarf virus, is a disease that's gaining in prominence, and one of the main reasons is because of our milder winters. So what we're seeing is the aphid down here in the bottom left. They would typically die off during the winter, but what we're seeing is populations are tending to overwinter and there's carryover, and then we get the infection and the yellowing of the leaves. Again, another important disease that can have significant impacts on yield potential of any crop. More recently, grass weeds have become a really significant and pernicious problem in the tillage sector. And there was an excellent conference held in Killashee a couple of weeks ago, wrapping up what was called the ECT project, where farmers were very honest about some of the problems they have had with diseases such as blackgrass, as you can see here, and called blackgrass for the obvious reason that the head is dominantly black in color. But each one of these plants can produce up to 6,000 viable seeds. So very, very quickly, it can rapidly multiply through a field if the field isn't managed properly, if the farmer doesn't adapt their cultural or indeed their herbicide actions to control it. And it is really a significant problem that we have, which requires a zero tolerance. We look to the UK and we see the situation that they've had there, where that zero tolerance approach wasn't possibly adapted as quickly or as detailed as it should be, and it's led to significant challenges for several farmers. One of the challenges then, of course, with blackgrass, and these are data shared by one of my colleagues, VJ in Oak Park, is that you see here that this is the impact of herbicide resistance or tolerance on blackgrass populations. And what you would hope to see when a farmer sprays herbicide, whether it's an ALS type active or an ACCKs, is that you would expect to see this scenario down here, So these are two populations, one from Ireland of blackgrass and one from the UK, which are sensitive to herbicides. And we know they're sensitive because obviously they're growing here in the control, treated with water. And over here, what we have is 100% plant death. What's very stark and very obvious, as you'll see now, of course, is that as you move up, we have populations from Waterford, 2 from Meath and 1 from Cork, growing happy out in the absence of the herbicide, but unfortunately in the presence of herbicide, they're growing quite well. And that's not good. So what it does is it highlights even further the need to ensure monitoring, absolute vigilance, zero tolerance approach, and be able to adapt your cultural systems and your management systems. Thankfully, we have Vijay and his team are monitoring this closely and accept samples from farmers so that they can give farmers back the herbicide profile of the seed that they collect. But when you package all of this together, what you have is a situation where all of these could be occurring in the same season. So whether it's disease incidents in the top, whether you have a drought spell like we had for the last couple of years, typically late spring, early summer. Obviously land availability, we all know the issues that we have with that at the moment, nutrient use, but ultimately the key thing is maintaining profitability. And that's becoming very, very difficult. And it's difficult now. And of course, we have to look out how difficult is it even going to be out in the next 20, 30 years when we have climate impact being much more significant than it is based on model predictions. Of course, there's one thing to talk about climate impact, but, and Helen mentioned it as well, we have farm to fork ambitions, which are the policy drivers. So we are all fully aware of farm to fork and what the ambitious goals are. And the whole foundation of farm to fork from a cropping perspective is built on the pyramid of IPM, or integrated pest management. And as you see here on the right hand side, the foundation of that integrated pest management is rotation and crop choice. And then obviously cultural issues such as time of planting, exploring biological control, monitoring evaluation of diseases. And then the last thing in terms of controlling diseases is, if required, an application of a pesticide or a fungicide or herbicide or whatever. But the goal of farm to fork is to try and to reduce the dependency at this part of the top of the pyramid and to focus more on the bottom. But it's not a coincidence really that when we look at the bottom here, what we've got is crop choice is key, because it doesn't really matter what the driver is going to be, whether it's a policy driver or a climate driver. Crop choice is what's going to be key, and fundamentally it's variety choice is the most significant step in any crop production system. So how do we increase crop resilience to stresses, whether they're abiotic stresses or biotic stresses? So this is a nice drone shot of our annual trials in Oak Park, where we're looking at thousands of research plots every year with the work of the team there. And the key thing is, what we see is, working with breeding companies and stakeholders, is that to breed a variety, obviously, as we know, it takes a lot of time. The average time for a cereal variety is about 10 years, for a potato variety is up to 12 years. And this simple schematic encapsulates it well, because you're crossing two parents, A and B. One has one trait that you want and the other has the other trait. You're doing it by hand, you're getting seed, you're growing that seed out in a field, and you're getting that diversity that's out there. And that's key to any breeding program. Diversity is key. And assuming that it's adequate and that you've adequate parental material available, then you'll generate a stepwise increase in yield or quality or whatever trait you're looking for. But what if that is not the case? What if the parental material is not going to be as robust and does not have the resilience traits that we need to address what we predict will happen in the next 20, 30, 40 years that are out there? Bearing in mind, of course, that it takes so long to produce a new variety. So what I'm getting at is really breeding techniques and new genomic techniques, or what are called NGTs. So while it can take up to 12 years to breed a new potato variety using approaches such as what's called cisgenics, you can accelerate that breeding process. And what you're doing then is you're transferring a gene from a wild potato into a conventional potato. Now, of course, that can be done through conventional means, basically taking pollen from a flower and transferring it in, getting seed, etc., out here to your diversity. But It's been shown for varieties such as Bionica and Toluca, when you transfer obviously in from a wild species through conventional means, you're transferring in, you're mixing up all those genes from the wild species as well. So you may indeed get the resilience trait you want, but you're going to get a pile of traits that you don't want as well. So using techniques such as cisgenics, you can be much more targeted in that approach. The other form of NGTs is gene editing, which obviously has captured a lot of the popular press. And editing is really about editing or disrupting what is already there. So you're not adding in any new DNA into the target variety. And both cisgenics and editing can provide an opportunity to enhance what we already have. So in effect, as is written here, you're looking at the development of a variety version 2.0. So you could have an excellent, could be even a grass variety, could be a clover variety or a cereal variety, potato variety. It ticks all the boxes in terms of digestibility or quality or food processing, but it might be falling down on an agronomic trait that needs improvement. So you can go in with editing, and the potential is there with the tool, because that's all it is a tool to improve that agronomic potential of the variety. So just to give you a schematic of what it is, editing is the term is termed CRISPR, which is an acronym for a very long term that's written there, but it's based on the use of specific proteins, and those proteins will make targeted cuts along the strand of the DNA. And you can guide those molecular scissors with effectively a molecular sat-nav. And it can be very, very specific and very targeted and very precise. And I'll show a few examples in a few minutes of what it can deliver. Cisgenics, on the other hand, is where you put in a piece of DNA that comes from a related species. So as I said, wild potato to conventional potato, wild apple to conventional apple, etc. And just as a comparison or as a contrast, this is the opposite to what we would traditionally believe to be GMOs. And indeed, editing and cisgenics are legally defined as GMOs based on the European Court of Justice finding of 2018. But when you're talking about the traditional GMO, that would be like a GM maize or a GM soya. That's what's called a transgenic. So you're taking a gene from a different species, in some of those cases a bacteria, and you're putting it into a plant. So you could take a gene from a daffodil and put it into a wheat plant. That's a transgenic approach, and obviously that's never going to happen naturally. So why am I talking about this? Well, one of the reasons is because in July of this year, the European Commission published a new proposal on plants obtained using NGTs, specifically cisgenics and editing. And when I mentioned earlier that the Court of Justice published their findings in 2018, they said that these techniques were grouped under GMO legislation. And that created a couple of challenges for the Commission. And I suppose in the context of farm to fork and other food security issues that have come through more recently in terms of Ukraine, the Commission has been working with several different agencies, the Joint Research Centre and other institutions, trying to determine and delineate the impact, potential risk or impact of plants derived via editing. And they've published this proposal as a way to, I suppose, map a path forward in terms of regulating them and developing them, because they have said themselves, using their own language, that the current regulatory system around GMOs is not fit for purpose. And what this means is that you can have the option, or I suppose the potential, of materials such as what we have here, these cisgenically engineered potatoes, could maybe make it out into the marketplace in the years ahead. So this is work we did several years ago, actually 10 years ago, on a project called the Amiga project, which was funded through the EU. And we were working with colleagues in Wageningen University, and what we were doing was we were evaluating an impact assessment of engineered disease-resistant potato, basically potato that had been engineered for late blight resistance, using a variety called Desiree, which is very poor late blight resistance. And as you can see here in the plots on the left-hand side, looking very poor. But when that particular variety was complemented with a gene from a wild potato species, you can see the difference here on the right-hand side. So it developed very strong resistance to late blight for 3 successive seasons. And as a result, the spray requirement went from an average spray of 13 sprays for a standard Desiree crop down to a single spray as a disease mitigation measure. So the environmental impact obviously was dramatically reduced. and obviously yield was preserved, and it demonstrated the role of cisgenics as a way to enhance an existing variety. And this work was included in the JRC report, which ultimately led into the European Commission proposal that was produced in July. So to give you some other ideas of what editing or cisgenics can do, I pulled out a few examples from the literature, and just to, I won't get into the heavy detail, but this is an example of where within wheat there are 3 copies of every gene. And what this group did, Abe et al., a couple of years ago, was that they were able to edit each individual gene that controlled dormancy. So as a result, they were able to control dormancy in wheat, as a result preventing pre-sprouting, which under very wet climates and which would prevent farmers from getting in and harvesting their field, could actually be an important trait into the future. Powdery mildew resistance is an important trait for wheat cultivation around the world, and can be a problem here in Ireland, not as significant as obviously the likes of Septoria or Fusarium. But what they showed here in this work by Wang et al., again, this is over 11 years old, is that on the left-hand side, this is a wild-type leaf covered in powdery mildew pustules. And when they were able to edit the 3 copies of one gene, they got complete resistance to powdery mildew on the right-hand side. When we move to a more dicot system, oilseed rape, this group, Zheng et al., what they showed was that compared to the control oilseed rape, which is quite tall in stature, they were able to edit 2 individual genes. And what they were able to do was reduce the stature of the crop itself to smaller, probably about 20% smaller, but actually they increased yield by approximately 20% as well. So it's what we're seeing here is that editing, when it's applied to so many crops at this stage and so many species, not just in tillage, but horticulture and forestry as well, is that it's really lifting the lid and the capacity to tweak and deliver new traits. So some of the crops I've listed here, and an awful lot of work has gone into rice, for obvious reasons. It's such an important crop, but it's not just disease resistance that I talked about on potato. It's climate stress and quality enhancement. And this last example here from Sanchez Leon, where they actually went in and they edited over 30 individual genes to deliver a much reduced gluten wheat using editing techniques as well. So looking ahead, just to give you an idea, in Europe, there's an awful lot going on in terms of this proposal. It was published in July. The legislative process is underway with the Council and the Parliament, a lot of discussions and obviously tweaking and other things I'm sure are going on. But it does require a vote by the European Parliament. And it's possibly anticipated that could happen before April 24. Or failing that, it would be after the election, so there'd be a delay. The important point to make, though, is that outside of the EU, varietal development using NGTs is continuing at pace, because the likes of the UK and China, USA and elsewhere, they see it as a very valuable tool. And indeed, that's what it is. It is an important tool. It's been demonstrated that it can deliver material with increased resilience. And looking ahead, when you see the challenges that we have in terms of climate adaptation, what we're going to have to do, we're going to have to explore the use of all the techniques and technologies that are out there. So while we have this version of editing now, I have no doubt that in 3 and 5 years' time, we'll have another version and there'll be another advancement. And all of these things are indeed going to be very important. So that's it, Siobhan. I'll leave it there, and I'll be happy to take any questions.
Super, thanks a million, Ewan. That was very interesting, and I think gene editing is a topic that people are very confused on. I think in general, people in agriculture think it's a good thing, but we don't really know what it's about sometimes. Like you described there, you said it could take 12 years to get a conventional variety to market. So I'm just thinking on potatoes. So if it takes 12 years for a conventional variety to get to market, is it going to be fast enough to keep up with blight and resistance to fungicides? You know, are we going too slow in our breeding at the minute if gene editing doesn't come through?
I think there's a real risk of that, yeah. Because I mean, you have to look at just the 2023 season. You know, we had a pretty cool springtime, bit damp, and then we had a drought. and then we had rain, and we had multiple seasons in one season. So adaptation and resilience in one season for a variety is a huge challenge. So equipping varieties using conventional techniques, yes, it can be done. Will it take a lot of time? It'll take a huge amount of time. And I think when you look at a technique such as editing, you don't have to reinvent the wheel. You can take an existing variety that, you know, meets all the processing traits and requirements required. And potato is a great example. There are so many quality boxes to be ticked. When you start breeding in new varieties together, you know, it's a huge amount of effort to attain what's required for processors. And as we know in Teagasc, we've been breeding potatoes for 50 years, so we know what it's like. So I think with anything, Siobhan, when you're faced with a huge amount of problems and challenges, you want to know that you have all of the tools available on the table to help you get through them.
I want to go into a little bit more detail now, and forgive me for this if I sound silly, but I'm sure other people are thinking it as well if I'm thinking it. When you talk about snipping the genes and making cuts to the genes, so I always hear that gene editing is very similar to conventional breeding. You can't tell the difference between them. So what are you actually doing when you're snipping the gene? Are you getting rid of something that's making the plant you know, more prone to disease or whatever it may be?
No, a really good question. So you're right. So editing in its simplest form, and of course there's various different versions of editing, but editing in its simplest form is indeed, as you said, you're snipping a gene. And when you cut the sequence of a gene, you can effectively turn off the gene. So it's a bit like a light switch for a bulb. And editing as a technique has evolved so much in the last 5 years. Actually, the analogy of a light switch and a bulb works well, because you could put a dimmer on a light bulb, and that'll turn it up and down slowly, and you can tweak that. And indeed, using editing, that's exactly what you can do. You can turn up the activity of a gene, you can turn it down, you can turn it off. So that's what it will do. Your point about it being equivalent to conventional systems is one of the reasons why the Commission probably struggled with this so much, because with editing in its simple form, when you make a single cut, you introduce what we call a snip, or a slight little change in the code. But that happens naturally. That's evolution, constantly evolving and changing sequence to try and find the best variant or the best trait that will emerge. So when you make those changes naturally, or if you make it through editing, you can't tell the difference, because there's no footprint left to say this was made by editing or this was made by Mother Nature. And from a regulatory perspective, if you can't trace it, how can you regulate it for it? And this was one of the conundrums that the Commission faced.
Will that be a reason why, if the legislation comes in to allow gene editing be used, will that be one of the reasons why it will come in?
It's been one of the drivers, yes, definitely. And I mean, a huge amount of work has gone into looking at ways to improve the traceability of it. But this new proposal is what it's proposing is that up to 20 modifications could be made, and that edited variety or cisgenic variety would be classified as a conventional variety. So it's just a standard conventional variety that goes into the standard system that we have at the moment. But that opens up a huge range of possibilities, because under the definition of 20 modification, it would mean you could add in 12 cis genes. So you could add in 12 wild potato genes to confer resistance to multiple different things. And you could make 8 other edits to improve an abiotic stress resilience. So it gives you the option then to stack the traits that you want. And that's the key thing.
Okay. We'll give you a break for a minute, Ewan. We'll bring in
problem.
Helen. Helen, I have a question in here from George Ramsbottom. Is clover the main contributor in the mixed swards to dry matter yield through N fixation and to animal performance in the mixed swards? And just before you answer that, Helen, you can be thinking, please do put in your questions into the Q&A tab if you have them.
Grand. Yeah, certainly look at clover is key in terms of the engine for production of those swards, because we've reduced down the N input so much that obviously you still need nutrients in the system, and the clover is fixing that nitrogen and making it available. So from that perspective, yes, absolutely, the clover is key to those swards and their performance. But I suppose when we look at how the multi-species swards are compared to the ryegrass white clover swards, and some of the things that I was looking at, some of the traits, I suppose, or functions I was looking at there, we did see that we have lower nitrous oxide emissions associated with the mixtures per ton of herbage that's produced. We saw that there's increased environmental resilience, so increased resilience to environmental stresses like drought when we have the herbs in there. And then again, in addition to that, the animal health benefits are associated with having the herbs in the swards as well. So certainly from a production perspective, but even at that, you know, it's definitely the clover is very important. And we need at least about 30% of dry matter coming from the clover. But, you know, we saw increased yields, herbage dry matter yields from our multi-species relative to our ryegrass white clover swards.
Is some of that the improvement in soil health, Helen? Is it the impact of drought or water infiltration, or just a build-up in carbon maybe, or exudates or something?
Look, it's probably going to be a combination of all of these over time. The carbon story is a much longer term thing than we've been able to look at yet. I mean, we've taken kind of baseline samples, but we're probably talking, well, we may look next year, will be 5 years in. At that stage, we might begin to see something, but we're really talking probably 10 years plus before you start getting anything really significant from that.
You have the baseline samples taken anyway, so okay. Helen, like, so the department has come out with their multi-species sward scheme, and it's got fairly good uptake, but I hear from farmers, they're asking me, you know, they put in their multi-species sward and it's not good. They're disappointed with it.
Okay.
Their swards they're good, like multi-species swards need good P and K levels. They need a proper pH. Is there a bit of a danger at the minute, because you've shown us all the benefits of multi-species swards today, but is there a danger that people put these seeds into the wrong soil, they have a bad experience and people are turned off? Does there need to be more education around how to plant these swards and where to plant them?
Yeah, well, I guess really, you know, when you're talking about establishing a multi-species sward, you're using the same kind of rules as establishing a ryegrass monoculture, in the sense that you need to have good soil pH if you're going to have good legume establishment. And as I said, legumes and nitrogen fixation through the legumes is key to the performance of the swards in terms of yields. And then, you know, P's and K's, I guess, we're a bit less sure, but we've been targeting at index 3 for both of those. So you're not talking about going down to the bottoms, if you want to call it that, putting them in there and hoping for the best. These are, you know, it's an improved grassland type, and it's supposed to, you know, you need good soil fertility to further performance and good soil pH. Yeah.
Very good. Brian Buckley says, great presentation. Have you looked at these mixes in silage making and their fermentation characteristics?
Yeah, Thomas Maloney did a PhD on that in Teagasc Grange with himself and Padraig O'Kiley. So certainly, I suppose, just to summarise, because Thomas had an awful lot of very detailed results from that, but I suppose overall what he found was that, look, you can make a good silage from these swards, there's no problem, but if you have them under bad conditions, and we found this ourselves in work at Lyons as well, if weather conditions are not good, it's very wet, then you're going to end up with difficulties there, because there's a higher moisture content associated with the herbs anyhow, and particularly in we'd say the year after reseeding, where you've got a high herb component of the sward, it can be a little challenging, but you just need to give them a longer wilting time, and then they can make a really good silage. And I mean, we're seeing that coming through in the animal performance, because the animal animals are being fed, they're being over the winter period on the silage that's coming from their respective sward type, and that work that I was talking to you from Lyons. So the animals are performing and we can make a really good silage, but it is a little more challenging if you've got wet conditions coupled with a very high herb content. So yeah, certainly a need for greater wilting, and they probably don't lend themselves well to a second cut system. You know, you're probably talking early year
Yeah.
when you can get
You need the good weather anyway for the wilt. Ewen,
with the high herbs, yeah.
okay. Ewen, last question to you. We hear about IPM. You mentioned IPM in your presentation. Really important on tillage farms. I feel tillage farmers are very aware of IPM. They're good at implementing it.
Yeah.
So if we are to see a real reduction in our pesticide use in time, do we need to go to gene editing, where maybe these plants will eliminate the need for fungicides or herbicides or whatever it may be?
Yeah, I think if you look at it the opposite way, if you don't go to techniques like editing or cisgenics, if we don't consider them as a breeding tool, I honestly can't see how the targets can be met, because it starts with the variety, as I said at the start, you know, and the variety needs to have the capacity to resist. It'll need help, and that's where you have the other steps in an IPM pyramid. But it doesn't matter what the stress, but if the variety isn't equipped to deal with it, it's going to be very, very difficult.
Okay. Thanks very much, Ewen, and thanks to Helen. Thanks to both our speakers today for excellent presentations. Thanks to the ASA for organising and hosting. This is just the first webinar in this series. So on Thursday at 1 p.m. on Zoom again, Tommy Boland will be joined by Pat Toohey of Teagasc to talk about drainage and peat soils and rewetting. So definitely a topical issue for Thursday. So you're registered for that if you're registered today. So tune in and see what's happening on it. Thanks very much.
Thanks.
Thank you. Bye.
Thanks.